All right, we are recording. How do we get his slides projecting them? Or
They are
transmitting or?
share screen.
No, you're right. You got it.
I know, but it flew off.
Okay. And then let's try to
Should work from here.
I don't know. We'll ask people to mute.
Dante?
Yes, ma'am. It's not advancing.
Okay, so I'm not sharing the right screen.
So try advancing. Oh,
Yeah,
that's a good. Okay, then go to, go to,
It's
go
not
to,
the problem.
go to the presentation.
It's not the problem. It's
Oh, that's not it.
share screen.
all right.
It should be.
Okay, now go ahead and hit down here, the reason that this is not advancing. How strange. I don't know how to get that to advance so that they can see it. It's doing it on your computer, but it's not doing with the live share. It's okay. We'll figure it out.
I may just have shared with the wrong screen.
Problem is you can't get it into slide mode here. That's really strange. You could open it back up again, close it and open it if you wanted.
Yeah, it does, doesn't it?
Now, here's the thing, can we manually advance through this? Because that would make it really easy.
No, 'cause
Wow.
it's
it
not
won't let us
sharing.
advance
Because
at
we're
all.
not sharing.
Okay.
Wait a minute. What did you do?
Yeah, you had some.
Oh, no.
Okay, great, all right.
Okay,
So
so it-
Can everybody see that? Yeah, I see
I'll
it.
just have you
Dante,
advance.
it's up
Got it.
here.
I got it.
Perfect.
Yeah,
And,
it.
oh, you know what? Look at this. Let me do it from here now.
Okay.
Perfect.
Can everyone out there see the presentation now? Give me thumbs up. Give me thumbs up if you can see the presentation. You cannot see the presentation?
See you with two screens on either side of you.
Say again?
We see you from the camera, we see your face with two screens on either side of you.
Okay, because we're not sharing the right thing.
Basically, we're not seeing the presentation, no.
No.
Okay, Chris. Okay,
There we go.
got it?
Okay,
Okay.
now all you need to do is get in presentation mode because you're now in edit mode.
So go in. We're down there.
Doesn't seem to work, which is why we were doing it the other way.
I'll tell you what, if we
Do you-
just
did you s-
manually go through, that's great, as long as they can see.
Can you see what, a river, a nice picture of a river?
Yes, nice picture of a river, beautiful rock. with
Okay.
moss.
Yes.
Or good enough.
Okay.
Yeah.
Yeah.
All right,
Good enough.
we're good to go, guys.
And
Want to introduce our speaker now?
yes, we are
Okay.
recording.
Yes.
Okay,
Fair enough.
okay. Please, I would like to ask, we are recording not to ask the speaker any questions during the presentation. And if you will put them in the area where you can write questions, we'll make sure that they all get asked afterwards. Okay. The I'm announcing.
Checking to see if we're recording.
I don't see the recording icon on there. Okay, it is recording.
Okay. Tonight, our speaker is Danté Fenolio. He is the head of San Antonio Zoo's Center for Conservation and Research. He is an amphibian specialist and has done a lot of work in different, everything from aquifers to ponds to lakes, to the ocean, to rainforests. And so this, I think, will be just a fascinating lecture for us. And of course, we have our own organisms that we look for, like the Texas blind salamander and all of those that are specific only to this area here in the San Antonio Edwards Aquifer. So I think this should be very, very interesting. The zoo also has other projects that they work on all over the world, everywhere from Peru, Chile, China, Japan, all of this working with different animals that are in these water and aquatic habitats. So Dr. Fenolio is from the Santa Cruz area of California. He went to the University of California at Santa Cruz, where he got a degree in ecology and biology, and then a PhD from the University of Florida in Miami in biology. He also did some work for the Botanical Center in Atlanta, working on a database of the amphibians. So we have someone here tonight who can tell us many things. He has taken all of the photographs here, which, as you can see, are gorgeous. So let's welcome Dr. Danté Fenolio. Oh, no, I didn't read it.
It's okay. I got it.
Where'd you
Okay.
get that
Can everybody online hear me? Oh, that's right. The speaker's not.
No, they should be able to respond.
Yes, I can hear you.
Okay,
Yes,
great.
I can hear you too.
I might be able to forward. Sorry, guys. We're working through a little bit more technical difficulties. I'm trying to figure out how to forward these things manually. Just give me a second. Yeah, the problem is it's not. There we go. Ish. No,
Click on it. Yeah.
there we go. All right, I think we're good now. Okay, so thank you guys for coming tonight. I appreciate. Sorry about the technical difficulties. So all I'm basically going to do tonight is talk about our conservation work at San Antonio Zoo's Center for Conservation and Research. And I hope that I will convince you that it is diverse. Where I'd like to start all of these seminars, particularly when I'm in front of university students, I want you to clear your mind for a second, and I want you to think about a pristine place in nature. It doesn't matter where that is, you pick, but somewhere where you think it's still a wild place. It is not impacted by humans. And then I want to come back to reality and discuss the sort of the harsh realities of what's really going on around the globe. And unfortunately, what's really going on around the globe is the fact that there are not corners of the planet that are not impacted by humans any longer. Even if we go to the poles and we take ice samples or ice cores, you will find pesticides. So the sad truth is we're in a time and a place where humanity's indelible fingerprint is pretty much all over everything. With that in mind, as a conservation biologist and in line with all the rest of the many conservation biologists out there, it is incumbent on us to fight for what is left. I threw this slide in because it's really sort of an important and emblematic image for what we do at the Center for Conservation and Research at San Antonio Zoo. And really, we mostly work with non-charismatic species. So when we talk about charismatic species, when we talk about pandas or wolves or grizzly bears or polar bears, sea turtles, those are things that are relatively easy to raise conservation money for. The sad truth is the vast majority of species that need our assistance are not things that are easy to raise conservation money for. Things like this little venomous viper that is now probably found in just a few mountaintops in the highlands of Mexico in some cloud forests. That's all that's left, but you can well imagine the difficulty that we have raising money for a little green viper, right? It's not like raising monies for pandas or bears, etc. So that's kind of the theme for tonight's presentation. But in general, what I'm going to do is I'm going to talk to you about contemporary zoos and a future for wildlife from the perspective of a zoo, talk to you about the diverse conservation work that we undertake at San Antonio Zoo, why so many amphibian projects. That's kind of a thing, and I'll talk about that. But in conjunction with that, I need to talk a little bit about amphibian declines and the forces behind them, how I believe one institution can actually have an impact. And then I'll give you some examples of specific amphibian conservation projects that we're involved with, and then why you should care. I always like to end my talks with why the average person out there, even somebody who is not environmentally minded, should really care. And I think that those are important points to hand out. So when we talk about contemporary zoos, one of the things that I like to point out is that they're perfectly positioned to deliver conservation messages to the public. Why do I say that? Zoos and aquariums around the world see over 800 million people annually. That's more than all of the professional sporting events in the United States combined in a given year. So more than all the football games, all the basketball games, all of the softball games, baseball games, all of it combined, more people annually go to zoos and aquariums around the world. So it's a huge interface, right? So zoos and aquariums become key and critical stakeholders, right? And pieces to that puzzle because they have such a massive interface with the general public. And in as much, when families go to a zoo or an aquarium, what they're doing is they're going to entertain their families, right? And so they've designated monies in their budget to take the family out for some fun, right? So these are entertainment dollars, and zoos and aquariums are the only major stakeholder at the table that can reroute those monies into conservation. And so that eliminates one of the entire arguments that anti-zoo and aquarium folks often bring up. And that argument is zoos and aquariums compete with places like Nature Conservancy, Sierra Club, and others for conservation money. And in fact, it's an additive effect, and there's virtually no competition between them. In fact, I would argue that some of our key and critical partners are exactly those institutions with regard to the conservation projects that we've gotten involved in. So again, I don't believe that there's any major competition with any of those agencies. Sadly, the last place where you're going to have genetic diversity for some species are zoos and aquariums. And on a growing basis, this is becoming more and more true. Zoos and aquariums are actually holding far more genetic diversity inside of their walls than exists in the wild, with a growing list of species. Never forget that zoos and aquariums offer specialized facilities and trained staff. That is a huge characteristic. And again, it's one unique to zoos and aquariums. And zoos and aquariums are working on well-developed education programs. I'm very proud of the education department that we have at San Antonio Zoo and the massive impact they have on children of all ages. Don't underestimate the long history of zoos and aquariums. There is a long list of species that would have vanished and only exist now in the pages of books on extinction if zoos and aquariums would not have waded in and gotten involved. And the truth is, something as emblematic as the American bison was saved by a zoo. The very last herd was purchased and protected by the New York Zoological Society many years ago. So I want to get into this talk by giving you an example of some of the diverse conservation programs with which we are involved, but I want to acknowledge that long before I showed up at San Antonio Zoo, the zoo was very famous for some of its conservation work. For example, the reptile department is internationally renowned for breeding critically endangered reptiles and amphibians. That program has been in place long before I was involved with the zoo. And the zoo is also very famous for its bird program. And there are a host of critically endangered birds that we work with, things like Guam rails and Guam kingfishers, whooping cranes, et cetera. And all of that work existed long before I arrived at San Antonio Zoo. So the first project that I'm going to use as an example is the deep sea research that we do in the Gulf of Mexico. And really, this project, it's been funded now for about 10 years, a little bit longer. It is a response to the Deepwater Horizon oil spill. That was one of the worst oil spills in human history. It was a very deep water event. And unfortunately, that oil spilled for many, many days before the wellhead was capped. And that oil continues to this day to impact the deep water community in the Gulf of Mexico, because one thing that most people don't realize, under pressure, oil doesn't float on water. So the bulk majority of the oil, the cloud of oil, is still there about 50 or to 100 meters up off the seafloor. Oh, wow. Okay, so we're not advancing through the images for the folks. Click over there. All right. Hit left. There we go. Yeah, it's not giving me any opportunity there to do anything different. I'll get out of your way.
What do you folks see? People?
I see the PowerPoint screen. And but
What
it
image,
looks like it's in edit mode.
what image do you see?
The river rocks, the original one, the second slide in the deck of slides.
Okay.
So if you, whatever computer you're on, if you're seeing the same thing that we're seeing, there you go. Now I see a cursor move. If you go to the very top left-hand corner, the fourth icon from the left,
Just a minute.
left, you're at the right.
No,
Go
we have,
left.
I know, I just have to get it so that it's visible. The corner is visible. It's,
Oh, right, yeah, so you're not seeing everything.
okay.
That is
Fourth
a bit
icon from the left.
Yeah, right there. You got, click that and click on slideshow.
Yeah, it doesn't seem to work. It's the
It
problem.
doesn't, does it? There's a little pull down arrow for slideshow, or click on resume
Okay.
slideshow.
Yeah, it doesn't seem to work.
That's funky. Oh, well, sorry about that.
What do you see now? What image do you see now?
I see a grass fire.
Oh, you do? That's what you should see.
Oh, yeah, it's not grass.
What do you see now?
I see a forested hill with half of it denuded.
Perfect. But here's how you control, Dante.
They're seeing the different, these are
Yeah, you need
going
to,
forward
I know,
slower
Well-
than he's talking.
they're seeing this,
Mhm,
So
not this.
right. I have no idea. This is
So
not my
you can
setup.
You can
No idea.
control it here.
Okay. Okay, so you want me to advance by clicking on the
Yeah,
left
try
hand
that.
margin? I will do that. No problem.
But you guys aren't gonna be able to see it.
That's all right.
Let's see what happens when we do that. Okay, so can everybody online see the Deepwater Horizon rig on fire?
Yes.
Okay, cool. I'm just gonna manually advance by clicking on individual slides. So we're involved in this project in the Gulf of Mexico, where we're looking at the environmental toxicology of the oil spill. and really the gross impact that the oil spill had on the pelagic community. And when I use the word pelagic, what I mean is that this is the community of organisms living in the water column, not down on the bottom, but in the water column. That is the pelagic community. And that community is really quite diverse. Yeah, so it won't advance up here. Okay. That includes a whole host of very interesting fishes, including things like deep sea angler fishes. One of the things that I've gotten the most out of, having been involved in really dozens of research cruises out to sea, is that over time, you get to see various developmental stages of the same species. And so you get to see an adult, you get to see a juvenile, you get to see a larval stage. All of those things are really interesting to me. And that's one of the benefits to repeated trips out to sea. Now, before I go any further, let me explain to you what we do. We have, from the ship, we have a computerized net, and it's actually 6 nets in one. It's called the mock nest system. And we can lower that below the ship. We drop it down to 1,500 meters depth. And we go ahead and we open one of the nets at that depth so that we know we're fishing at a discrete depth. At that way we can fish at that depth, then we can close that net. We can bring the net up to another depth, open that net, fish at that depth, so on and so forth. And the reason that is so important, we need to know exactly where the specimens come from, the specific depth, so we know who is impacted by this oil spill and who is not. So after we're done fishing with all of those nets and we haul those nets back up on board and we start going through the specimens, the deep sea community that we're investigating, one of my jobs on this research collaborative is that I take pictures of all of the specimens that come up, because a lot of these things are very poorly known. We don't know color and life. We don't know what a juvenile looks like, and whereas we know what an adult looks like, or vice versa. So I spend all of my hours out on the ship photographing things like deep sea squid. Here I'm showing a variety of different squid. All of these things are species that came up in the nets. Here's a squid, a larval squid with eye stalks. That's something quite spectacular, a little bit different and not that common coming up in these nets. Larval fishes are also things that commonly come up in the nets. Give me 1 second. Should I turn the laptop so that everybody here can see that?
Yeah,
Right. I don't think I'm going to disrupt anything if I just turn this laptop around. That way you can see what we're trying to do, folks, is just get it so that the folks here can actually see this too. Let's see where I can prop this up or not. Yeah, actually the HDMI cable is not long enough. All right. Okay. So back to larval fishes. So the spectacular thing for me as a biologist is seeing all of these different stages of all of these incredible pelagic species, most of them deep water species, and learning about them. This next slide depicts a larval eel. You may or may not know that there's over 150 species of eel that pass through their larval stages in the Gulf of Mexico. That makes the Gulf of Mexico a critical larval habitat region for eels anywhere on the planet. And that is actually one of the big projects with which I am involved, a catalog of all of the larval eels that pass through these stages in the water column in the Gulf of Mexico. And you'll note that these things are compressed like a penny, right? So they look like a leaf, they're translucent and compressed like a penny, and they'll go through several months to even several years in this larval stage. Then they pass through their first set of metamorphic stages, and they'll look more like an eel. Then they'll pass through several more stages and actually turn into whatever the adult eel looks like. But another thing that a lot of folks probably aren't aware of in marine ecosystems especially are the parasites, right? So you notice that this deep sea hatchet fish has this really strange animal attached to its side with long string-like structures coming out of the rear end of the animal. This is actually a parasitic copepod. And the parasites on some of these deep sea animals that come up are absolutely fantastic. This is another parasite that came up off of a wahoo. Absolutely otherworldly. It's not hard to look at pictures like this and see all kinds of sci-fi movies and where, you know, some of those ideas came from. But maybe the thing that allows us to promote STEM fields to kids in particular are the things that are big and toothy, right? So things like ogrefish on the left and telescope fish on the right, those are just really the spectacular deep water predators that the deep sea community is so famous for. This, of course, is a dragonfish with the barbel hanging off of its chin. At the end of that barbel, you notice a little dark structure. That structure glows in the dark. So one hypothesis is that structure is used to attract larval fish, or I'm sorry, other potential prey items within striking distance. But we're actually working on another hypothesis that would argue that some of these bioluminescent structures are actually they have to do with mate selection between males and females in the dark. So the jury's still really out on dragonfish and what their barbels are used for. These are just examples of other species. Hopefully the folks online are seeing the entire slide and not just the abbreviated slide. This isn't really showing me what you are seeing. Yeah, see, it's not opening the entire slide for them. That's okay. Most of the slides are not vertical format, so it ought to be fine. Anglerfishes are another big group that allows us to promote STEM fields to kids. Anglerfishes are wonderful because they have their first modified dorsal ray is a structure called an elysium. And then at the end of that is a big bulbous structure called an esca. And an esca is basically a condominium for bioluminescent bacteria. Anglerfish don't make their lures glow themselves. They can't. They have bioluminescent bacteria that they live in a symbiotic relationship with. In fact, the only place on Earth that you'll find these various species of bioluminescent bacteria, you can find them in the water column and you can find them in anglerfish esca and no place else on Earth. So these are highly developed structures that don't just accommodate the bioluminescent bacteria, but they also have light reflectors and diverters. They've got little flaps of tissue internally that can cover up the light that's coming out of them so that they don't produce any light if they don't want to. And then they can refract the light in various directions to make this lure look very, very different at points in time. So this is a highly specialized relationship and one with which we've had a lot of luck in getting kids excited about biology, particularly deep sea biology. This is one of the best lures I could possibly ask for to get little kids into science. And again, with big and toothy, there's other deep water adaptations that are spectacular. You're looking at a thing called a hammerjaw here with a greatly distended stomach. So this fish can actually eat a prey item that's larger than itself, and its stomach will stretch out to accommodate that prey item. There's a lot of research and interest into how the small fish subdued the larger prey item in the first place. But in this case, it's a squid. So you can see the eye, and even below the eye, you can see what's called an ocular photophore, or the structure below the squid's eye that glows in the dark, right through the very thin tissue of that fish's stomach. And you can see just how stretched out it actually is. The invertebrates in the deep sea community are no less fascinating. This is a giant deep sea amphipod with highly complicated eyes. Some of the deep water crustaceans are phenomenal with amazing colors and shapes and sizes. And when I talk about sizes, this is a deep sea isopod of the genus Bathynomus. This is a baby that you're looking at. They grow to the size of a bowling ball. So one of the cool things about deep sea wildlife is the ability to create light. And I think a lot of folks are not aware of the fact that bioluminescence, or the natural production of light, is the most common mechanism of communication on the planet. We have so few examples of it in the terrestrial environment that we tend to forget about it. But in the deep sea environment, up to 95% of all of the species present can actually generate their own light. And there's hundreds and hundreds or thousands of species in the deep sea environment. So again, it is the most common communication mechanism on the planet. So if you're looking at something like a firefly under normal light, and then you turn the lights out, this is what you see. Bioluminescence is used for a host of different reasons. In this case, it's something called counter-illumination, and you're only looking at the underside of the squid. This squid lives exactly at the zone where the faint light from the surface is just starting to disappear. If you're a predator and you're looking up towards the surface, it's very easy for small organisms to get silhouetted. And if a predator can see your silhouette, it can find you and it can eat you. So what a lot of fish and squid and shrimp do, they've got photophores, or light producing structures, on their ventral surface, their underside, and they produce faint blue light, and literally it makes them disappear in plain sight. It gets rid of their silhouette. Lots of things glow in the dark. There are a lot of gelatinous organisms that glow in the dark. If you're moving along the ship at night and you look in the bow and the wake, a lot of times you see bright flashes of light, and a lot of times that's things like jellyfish or cnidarians and such that are actually producing their own light. Light as an escape mechanism is another big one. This is a shrimp expelling a brightly glowing cloud of fluid that flashes brightly for just a second. The shrimp swims the other direction, and a potential predator is startled with that bright blue flash of light. We were talking about counter-illumination. This is an example of a fish with all of those photophores, or light-producing structures, on the underside, just like I had mentioned earlier, to get rid of the silhouette. Now, when I'm going through these examples, I always like to go back to education and outreach, because it's so important to the zoo to reach kids and make sure that we promote STEM fields to kids, particularly in the United States. The United States has fallen way behind in STEM fields, terribly behind. And so any hook we can use to bring kids into the fold is acceptable by me. And so one of the things I did in handling education and outreach for the Deep End Restore project, we came up with a cartoon character called Squirt the Squid. I hired an animation artist that would make 15 and 20-second short takes with Squirt, where Squirt would explain something like bioluminescence or big teeth or life in the depths, and sell that to kids through this animated character. So you're looking at Squirt the Squid right now, and this is one of the key tools that we use to get kids all over the world excited about the deep sea environment. And then one of the things we did is we made this website, and I know you probably can't see it because I see exactly where that's getting cut off. The website is deependconsortium, all one word, .org. And if you go there, there are free teaching modules for teachers to use at any grade level. And we had those developed and built specifically to be able to give out to educators all around the world so that kids could get excited about deep sea biology. The next project I'd like to talk about is one a little closer to home, involving Texas horned lizards. I have the good fortune of working with a biologist in my department, Dr. Andy Glusenkamp, who runs this program for San Antonio Zoo and the Center for Conservation and Research. And I was able to steal Andy away from the state of Texas. He used to work for Texas Parks and Wildlife, and he was the state reptile and amphibian biologist. So in that role and in that position, Andy realized very quickly how attached to Texas horned lizards the average Texan is. And so when I was able to bring him to San Antonio Zoo, he developed a really special program where we are breeding Texas horned lizards in the lab and learning how to put captive bred babies back out onto the landscape. And we're working with private landowners and foundations to fund this. But the whole idea is that landowners will work with conservation agencies and try to restore their land as similar as they possibly can, as close as they possibly can, to bring it back to what the land looked like before Europeans arrived. And a lot of times that involves opening back up the landscape. Humans like to let the trees grow in and shade things over. And if you're managing your land for quail, you are also managing your land for Texas horned lizards and keeping it opened up. You got to control feral cats, feral dogs, roads, no pesticide use, get rid of invasive plants, deal with fire ants, or at least try to manage fire ants. But if you can do that, we're willing to bring captive bred Texas horned lizards back out to your piece of property. And you might ask, well, why do folks need Texas horned lizards brought back out to their property? Texas horned lizards used to be virtually ubiquitous across the state of Texas, and they've ultimately vanished from about 40% of the state. So the species is not listed as a critically endangered species, but it is in decline. And we're really interested in seeing how you reintroduce captive bred babies to the landscape and how you can get them established. And Dr. Glusenkamp has done a wonderful job in getting this project going in the right direction. We've now gone through 3 releases on a rehabilitated piece of land, a private ranch. And in fact, the captive bred babies that we've put out there are taking and living and making it through the wintertime. And we've had a recapture, and we're also frequently finding lizard scat out there, lizard waste. And that is another real positive sign that what we're doing is working. All credit to Dr. Glusenkamp and the team at the Center for Conservation and Research for getting that to work properly. So the next project I'm going to talk to you about, and again, this is just to show you the diversity of conservation projects that we've got going at the Center for Conservation and Research. We are involved in subterranean biology in the Ozarks, and the Ozarks are an uplift in the central portion of the United States, partially overlapping states like Oklahoma, Arkansas, Missouri, and a tiny little southeastern corner of Kansas. In our work, we work with federal officials and state wildlife officials, and we basically go in and out of cave systems and document the biodiversity that's found in them. A lot of this is real basic entry-level biology where we are simply documenting the species that are present, the biodiversity in these subterranean systems. And right off the bat, you might ask why it is that we would spend the time looking at subterranean biodiversity. A lot of times, subterranean species are restricted to one or a handful of cave systems. They're some of the most range-restricted critters on the face of the planet. And that's something that needs to be paid attention to, because anytime you have a species that's range restricted, a human activity on the surface could really impact those populations in a negative way. So first, you need to document what species are where, distribution and diversity, and then you need to work on protective measures for things that have very restricted ranges. That's exactly what we've been doing with the Subterranean Biodiversity Project in the Ozarks for over 20 years now. Not surprisingly, that work entails some significant caving, including things like snorkeling in subterranean rivers, lakes, and streams. When we're in those systems, we're looking at all kinds of microhabitats, including spring runs, cave streams, drip pools, epikarst habitat, all kinds of specialized microhabitats, and looking for species that are common to them, but only found in those subterranean microhabitats. So I like to show folks what it is that we see in these habitats. And a lot of times, as a wildlife photographer, this is the kind of thing that I really like to try to capture, subterranean wildlife in cave streams, so folks get an idea of what it is we see when we're doing this work. So that includes things like cave crayfish and cave crayfish surveys. So one of the big jobs that we have when we work with federal and state wildlife officials, we keep tabs on how the populations are doing in several of these caves with extremely range limited species of cave crayfish. Again, that's work that we've been doing for over 20 years. A lot of times this work will introduce us to species that are new to science. You're looking at Oklahoma's first blind cave snail, That's an aquatic species, and we're in the process of describing that right now. But these subterranean communities include all kinds of really, really interesting creatures like pseudoscorpions and cave salamanders, and of course, bats. So I just want to throw out that bats are key and critical elements to a lot of these subterranean food webs. And here's why. In a subterranean environment, there's no light, therefore there's no photosynthesis. So you don't have plants at the base of any kind of food web because plants can't exist in the subterranean systems. What you do have are these things that academics term allochthonous energy inputs. And that just means that the energy originated somewhere else. So in the case of bats, they go out at night, they feed out in the surface environment, then they come back to a cave and they defecate in the cave, and they make things like guano piles. Those guano piles are the energy resource for a lot of these subterranean communities. And it's not just bats that do this. Cave crickets go and feed in the surface environment at night, and then they come back and they defecate and produce frass piles. There's all kinds of circumstances where organics can wash into subterranean habitats. But the point here is that it is a decomposition-based food web, and decomposition-based food webs have a lot less energy flowing through them than do surface food webs that are powered by photosynthesis directly. When you have a lot less energy flowing through a food web, you have a lot fewer individuals living in those communities and a lot fewer species involved in those communities. And again, that's another reason why subterranean fauna are so vulnerable to human activities. They tend to have small ranges, and in a given cave, there does not tend to be a ton of individuals or species involved in a community. So any little disturbance can push something towards extinction. Again, with each one of these projects, I want to emphasize education and outreach steps, because that is something that we live by at San Antonio Zoo. And in this case, we produced Cave Life of Oklahoma and Arkansas, which documents the subterranean fauna of the Ozarks. That was one of the first books with which I was involved. And really great experience where we were able to get into, well north of 500 caves over the course of that research. And we still do that research today. Now, an expansion of that research that's really, really been fun is the opportunity to work with imperiled cave fauna in the lab. And a lot of times we will have state wildlife agencies or federal wildlife agencies approach us at the Center for Conservation and Research, knowing that Dr. Glusenkamp and myself are specialists in subterranean fauna, and they'll say, hey, look, we want to know if you can develop captive husbandry and breeding protocols for this species that we consider imperiled, endangered, threatened, some level of threatened. And so we've got partnerships with all kinds of state wildlife agencies where we work with things like the Doherty Plain cave crayfish off to the left and the Georgia blind salamander to the right. We were the first institution on the planet to breed both of those species in captivity, and publishing those captive husbandry and breeding protocols is kind of the final deliverable that we are involved with, and then we'll move on to the next species. Dr. Glusenkamp has been managing a project where we are working with a blind catfish that is found primarily in an aquifer that lays beneath the border of Texas and Mexico. This is an imperiled species because there are things draining the water from that aquifer, and ultimately will bring it down, draw it down to such an extent that there won't be much habitat left for this species that's really only found in that aquifer. Again, I want to emphasize that subterranean species don't have anywhere else to go. They're obligate in the context that they're not found anywhere other than subterranean environments. A lot of times subterranean environments do not connect with one another. So these things tend to be found in very, very small ranges and only from a few systems. So they're very vulnerable to human activities. And just like the crayfish and the salamander that I mentioned before, we're busy trying to develop husbandry and breeding protocols for this outstanding blind fish that is endemic to said aquifer. In the process of working with these species in the lab, you get to see some very, very exciting firsts. This is the Doherty plain cave crayfish, a female with just a handful of eggs glued beneath her tail. That is the first female that's ever been bred in captivity right there. Likewise, we've got the Georgia blind salamander here. You've got a dorsal and a ventral view of the same animal. At the top in the ventral view, you can actually see the eggs through her abdomen. She laid her eggs, and once they hatched, this is what a baby Georgia blind salamander looks like just a few days after hatching. And again, that was the first time that imperiled species was ever bred in the lab. So I am very, very lucky to work with a group of people, the team at the Center for Conservation Research, who excel in the captive husbandry of a really sensitive subterranean species. And it's something that with which we're very, very proud. Our cave work does not stop in North America. We work with cave wildlife in Southeast Asia. Dr. Glusenkamp and I are both members of the Chinese Cavefish Working Group, which is an association of academic biologists that have all come together to better document the diversity and distribution of blind cavefishes across China. You may or may not know that China has a karst region or cave-bearing region larger than the state of Texas in the southern portion of that country. And China has more species of obligate groundwater fishes than any other country on the planet. It's at about 150 species and counting. Just like in North America, that cave work can be very challenging, but very, very exciting. And it's great to work with our Chinese collaborators from the Chinese Academy of Sciences and learn about these species in China that are just as vulnerable and just as unique. A lot of that work entails getting out into the various subterranean systems in South China and trapping the fish there so that we know what's present in some of those systems. And then a big part for the first 6 years that we did this work, we were funded by National Geographic. And what they were really after were pictures of the species in subterranean systems. And photographs like this of a blind cavefish in a cave stream became very, very important currency for us to keep that funding up. So all of these are photos. I had to teach myself underwater photography to do this. And there was a lot of time spent snorkeling in subterranean rivers and lakes in South China. But these species in Southeast Asia are quite spectacular. You might note that some of them have these odd structures protruding from their forehead. Again, one of these beautiful and magnificent biological mysteries. We have no idea really what those structures do. And it's a real point of study inside of our research group. We are not exclusive to China. We've worked in other areas of Southeast Asia, and things like the waterfall climbing loach from Thailand is another species that we've spent some time looking at. This is maybe one of the most cave adapted fishes on the planet. Believe it or not, this fish spends probably 80% of its life out of the water, climbing around waterfall spray zones and rapid spray zones on damp rocks. And it has these specialized fins for crawling around like a lizard, and it feeds on bacterial mats growing on the surface of the rocks. So this is another one of these crazy subterranean species that I feel very fortunate to have been able to see in the wild. And it's not just the fishes. There are all kinds of outstanding invertebrates. This is one of Southeast Asia's large house centipedes. For those living in Texas, you're probably familiar with a much smaller version of that. These guys are large enough where they feed on bats. The last project that I want to use as an example for the diversity of projects with which we're involved is one that involves the Amazon rainforest in South America. I was very fortunate as a child to have a biologist, a reptile and amphibian specialist, take me under his wing. And I spent the first 15 summers as high school wound down working for him in the Upper Amazon Basin. And a lot of that time was spent out on the lands of indigenous groups. I was able to make lifelong friends with a lot of these indigenous community members and learn from them. And there's nobody who can teach you more about the forest than the folks that live there. These forests are very special places, very complicated habitats that can be very visually confusing. But as a kid, this was what got me going and really drew me into biology. I've been very fortunate again to work with a lot of indigenous communities across South America, and some of these friendships have turned into conservation partnerships. This is a picture of myself with a couple indigenous community members from the Bora in Amazonian Peru. I grew up with these folks, and over many, many evenings and dinners, I wanted their perspective on why conservation wasn't working in the Amazon basin. Hundreds of millions of dollars have been spent, and there's very, very little to show for it. So I wanted their take on why it didn't work. I know what the Westerners have to say, but that's really not important. The folks that live in the forest are the ones that can inform me best. So these conversations led to a discussion where I came to understand these folks do fine without Westerners with regard to transportation, food, and housing. They've been in South America for a far greater period of time than Westerners have. Where they get into a bind is if they need Western medical attention or medicines, because cash is in short supply in the Amazon basin with regard to indigenous communities. So we began a discussion that was a long, winding road, but I wanted to know how it was that I might be able to support them with that sort of weak link in the chain, and that is providing cash when they need it. And they said one of the big problems is that a big conservation agency will show up and hand them big pots of money and say, don't cut down the forest that you live on, and ask them to sign a contract. And the big problem with that conceptually is it's not fair. We're superimposing Western concepts onto indigenous communities. There is not even a word for contract in the language that the Bora use. So it's really not a fair scenario. You know, as long as the money lasted, you know, there were great times had by all, but as soon as the money's gone, they don't understand the long-lasting repercussions of legal contracts. And it's really not a way to try to conserve Amazonian rainforests. So instead, what I wanted to do was a non-binding partnership with them. And we had more discussions, and they said, hey, look, what if we carved these little wooden animals out of balsa wood, which is a very light wood. Balsa trees are not endangered. They grow like weeds down there. A tree can go from a seedling to an adult in about 1.5 years. And we set up a program wherein San Antonio Zoo's Center for Conservation and Research, in partnership with other zoos like the Roger Williams Park Zoo in Rhode Island, we set up an office in the Amazonian city of Iquitos, hired a team, bought a boat to get our team out to these remote forest communities. And once a month, we will buy as many of these hand-carved animals as they want to sell us. So they can scale the income stream as they see fit. If they want to sell us 50, we'll buy 50. If they want to sell us 5,000, we'll buy 5,000. We ship those things back up to the United States and sell them in our gift stores. Predictably, the money from selling trinkets is not enough to support an office and a team and a boat in South America. So I really had to come up with some kind of mechanism to better fund such an expansive conservation initiative. And I asked myself what it was that we were trying to celebrate, and that's the biodiversity of these rainforests. And then I asked myself, is there an aspect of that biodiversity that really stands out? And of course, it's the freshwater fishes. There are more species of freshwater fishes in the upper Amazon basin than any other corner of the planet. We're at about 2,500 species and counting. Most taxonomists argue that by the time we're done describing everything, we'll be well north of 5,000 species of freshwater fish in these tributary rivers. So then I asked myself, okay, well, if we're trying to celebrate the biodiversity and a good emblem of that are the freshwater fishes, is there any other community on the planet that celebrates fish? And I immediately thought about Japan's fish printing process and fish print art that's known as gyutaku. And what we did is we built an art studio in our offices in this Amazonian city of Iquitos, and we contracted out with young Peruvian artists in that city. And I brought in gyutaku artists to train these young Peruvian artists in the Japanese fish printing art of gyutaku. So you see the late Heather Fortner there training some of our artists, showing them how to ink the fish and then how to press either paper or material fabric onto that fish, and how that entire process is done on various size fish. And then you end up with these outrageous exact replicas of these Amazonian fish. I threw this picture in because one of the things that we are very, very proud of, a lot of our artists are women. This is an aspect, a professional aspect for women in a corner of the world where there are not a lot of professional opportunities for women, and most of our artists are female. And they're outstanding, and they're producing things like these fish prints that you see here. Here are another couple examples. And we are able to take these fish prints, bring them back up to North America, make them available online, put them in art shows and such, and sell them to help offset the costs for the overall program. So that, in a nutshell, is a project that we call Project Selva. There are a lot more members than just the 2 zoos that are in this logo. I need to update this. Texas A&M San Antonio is part of it, and Moody Gardens has joined us, and the Snake Farm up I-35 has joined us. So we've got all kinds of partners from different backgrounds and fields joining us in this conservation project. And again, I really want to emphasize the whole reason we wade in and do all of this stuff. You got to get all the way back to the rainforests that exist on the traditional lands of these indigenous communities. And that's the biodiversity. And that's why the zoo is wading in, all the zoos and other institutions, to do this. We're worried about the biodiversity that can be found out in these forests. And that biodiversity is absolutely spectacular. The Amazon rainforest boasts some of the most biodiverse forests on the planet. I am director of research for the Amazon Canopy Walkway, which is one of the largest canopy walkways on the planet. And it stretches for nearly half a mile through rainforest trees and goes all the way from the forest floor on up to about 180 feet above the forest floor. It's a spectacular place to catch glimpses of Amazonian wildlife. And when we're talking about canopy wildlife, there's a lot of really special species there. There are canopy inhabiting lizards that are very specific to that microhabitat, and snakes just the same. That's what we're really after. And that's what I want to emphasize here, is that the biodiversity living in these forests has led us to work with the communities living in the forest. And I believe, I'm sorry, somebody's got their phone volume turned on. I believe that these forests and their biodiversity are absolutely worth wading in and fighting for. So again, I told you earlier in the talk that I would get back to why we have such a focus on amphibians. And I want to talk about that just for a second and start with global amphibian biodiversity. I don't want to assume that anybody knows a lot about amphibian biodiversity, but when we talk about amphibians, there are 3 living lineages. The frogs and the toads are probably the most commonly known and understood, and they come in all different shapes and sizes and colors and ecologies. They exhibit all kinds of bizarre behaviors, including parental care, where you have adults that will protect egg masses while they're developing. Then the second group are the salamanders and newts, just like the frogs. They come in all kinds of different shapes and sizes and colors, including blind groundwater inhabiting species right below our very feet here in San Antonio. And just like the frogs and toads, they exhibit all kinds of interesting behaviors, including migratory behavior. Then we get to the third living group of amphibians that people are far less familiar with, the caecilians. They have a backbone and a jaw filled with teeth, just like we do. They have no arms or legs. They're only found in the world's tropics, and they are predators. They typically eat things like earthworms. They spend most of their lives below ground, but just like the frogs and toads, newts and salamanders, you have all kinds of different ecologies and behaviors. One of them is really interesting. This species from West Africa, when it has babies, the babies live on the female for the first 6 months of their lives. They have special teeth, and they use them to rasp skin off of the female's back. And the
only food, in fact, that the babies feed on for that first larval period is the mom's skin. And she goes through a hormonal shift where she starts producing excess amounts of skin. And it's called skin feeding. It's one of the rather spectacular behaviors in this little known group of amphibians called the Sicilians. So when we talk about amphibians, there's well north now of 7,000 plus species that are known to exist. But when we talk about living amphibians, about a third of them, or a little bit more now, closer to 50% are in decline. Of the remaining 2/3, many of the species, we don't know if they're in decline or not. They're just data deficient. Some estimates suggest that as much as 200 species have already disappeared, and many of the declines have popped up rather suddenly and were recorded in the last 25 to 35 years. Now, I want to give you an idea of the size of this crisis. If you take all of the world's endangered amphibians and put them on one side of the scale, that is a greater number of species than all of the world's endangered mammals, all of the world's endangered birds, and all of the world's endangered fishes combined. So this is a serious problem. Amphibians need a lot of help. This is a brief breakdown of kind of our awareness of amphibian declines. I won't get into that right now, but the hypotheses behind the declines are multiple. Some of it unquestionably involves habitat alteration. 90% of all of the living species of amphibians that are in decline suffer from some level of habitat alteration, and that includes diverse human activities like putting in roads. Amphibians don't do well with roads. Harvest is another big problem, whether it's for food items, medicinal purposes, scientific collection, or even things such as the pet trade. Exotic species are another issue, whether the introduced exotic species is serving as a predator, as a competitor, or even as a toxic prey item. Ultraviolet light exposure may or may not be impacting amphibians. It's probably ozone related, mostly at higher altitudes, but the evidence is not entirely conclusive. Acidification of the environment is conclusive, and unfortunately, acid rain and acidified soil has really done a number on a lot of the world's salamanders. And then environmental contaminants, without question, things like pesticides, heavy metals, and fertilizers have done a number on amphibian populations. Emergent infectious disease has really been disastrous. Amphibian chytrid fungus has wiped out populations all around the globe. Probably humans have moved it around. Iridoviruses, also probably moved around by humans, have wiped out populations, and bacterial epidemics have really wrought havoc with a lot of amphibian populations. And then there's this business of synergistic interactions between these forces, where fertilizer combined with UV exposure leads to a far greater lethality to the amphibian population than either one of them would have done independently. Fertilizers and iridovirus, climate change and amphibian chytrid fungus, shrinking habitat, and water contamination. Those are all things that can work hand in hand and really do a number on amphibian populations. So you might ask yourself, if that's a global problem and it's pressing with so many species, how on earth can a single institution have a positive impact? And I'm going to pose three arguments that we have followed in our program that we believe we can make a positive impact in this global problem. And approach number one, we know we need more lab space. We know that we have so many critically endangered species that we're going to have protective custody over some of these things that are literally holding on by their fingernails. One of the things that my team and I specialize in are using shipping containers, used shipping containers, and turning them into laboratory space. This is a very cost-effective pathway to putting in lab space, and it also recycles shipping containers, which are readily available all over the planet. They can be moved on ships, on trucks, on trains, so they're mobile. And this is a very, very effective way to put in lab space. And in fact, we've been involved in either consulting or directly implementing these labs all over the world. It requires a little bit of equipment modification when you're setting them up, and a little bit in the way of specialized plumbing. But once you get it going, these shipping containers are highly effective laboratory space for smaller species like amphibians, invertebrates, and fishes. This is one of the labs that I was involved in implementing very early off at the Atlanta Botanical Garden. But this is absolutely a pathway where we can help develop capacity to work with critically endangered amphibian species in other countries. Approach number 2, I believe in training programs. And in fact, we bring in interns from all over the world to learn exactly what we're doing in our labs and spend some time with us. It's far easier to watch us working with amphibians and be there in person than for me to send them some sort of brochure and hope they pick it up from reading. So when we bring the interns in, we go over best husbandry practices and actually go through the breeding of an amphibian species. So hands-on training is, I believe, the best way to do this. And a lot of times that will involve how do you care for amphibians that are in the breeding process? How do you set up something known as a rain chamber, which simulates environmental changes in humidity inside and can get a lot of amphibian species going? Once you have the eggs, how do you manage them in captivity? How do you hatch those eggs out in captivity? How do you deal with all of the different foods that some of these tadpoles will require? There are as many feeding guilds of amphibians as there are of feeding guilds of invertebrates and fishes. A lot of these tadpoles have very, very different diets from one another. So we'll walk interns through that process. What happens when you're dealing with an amphibian that's going through the developmental process? What do you do with these larvae that are at metamorphosis, where they're changing from a tadpole to a frog? Once they make that change, how do you take care of them? And then what do you do with these species that go through all of their developmental stages within the egg? They're called directly developing species. There are quite a few of them from around the globe. And sometimes those need to be managed a little bit differently than the ones that go through a free living larval stage. So approach number 3, and this is a big one and a really important one that we hang our hat on: conservation partnerships built to last. And I'm going to give you 3 quick examples of those. The first one involves reticulate flatwood salamanders. And I was sitting at my desk years ago, and U.S. Fish and Wildlife called and they said, hey, we're dealing with a critically endangered salamander that we've never given permits out for folks to work with in captivity. This species used to range across a few states. Now we're down to a single healthy breeding population. And we think that we need to look at trying to breed them in the lab, or even having trouble collecting eggs and keeping them viable in the wild. So one of the things that I did, I jumped on a plane and I went out to the panhandle of Florida, where this species, the last stronghold of the species, exists, and went out and looked at the eggs in the field. And to show you how much of a geek I was as a kid, one of my hobbies or pastimes as a child growing up in the mountains of Santa Cruz, I went out and I collected salamander eggs and brought them home and taught myself how to hatch them in captivity. So oddly enough, that was a skill set that I am now using as an adult. Definitely one of those nerd things that came to fruition. And one of the things I remembered from my childhood, if you actually move the egg itself, a lot of times they don't hatch. Instead, you need to cut a divot, a dirt divot around the egg mass, take the whole divot so that you're not actually physically moving the egg itself. No problem. You can hatch those eggs out just fine. So as soon as I taught the folks that, we managed to go from 0% hatching rate of the eggs to about 99% hatching rate of the eggs. So that was an immediate impact. But this was a partnership at that point between U.S. Fish and Wildlife and the institution I worked for at the time. But the neat thing about this conservation partnership is that we were able to get universities involved and research biologists. Eglin Air Force Base is where this last healthy population exists. They have been incredible stewards of the land and have taken very, very good care of this patch of habitat where the salamanders are. They're partners in this. And then a whole group of other not-for-profit groups are partnered with us. So this is, when I say conservation partnerships built to last, I'm talking about making sure that you have different institutions that are committed at the institutional level, not the individual level, and ones that are going to stick by this project through thick and thin. And believe me, thick and thin is the real piece to this story, because it took us 10 years to figure out how to breed this species in captivity. But sure enough, we were the very first institution on the planet to breed this salamander in captivity, or even any salamanders related to it. And now we're working on breeding them in the lab such that any larva that we produce will be put back out onto the landscape in protected pieces of habitat. This is an adult looking at you. It's a very mysterious species. They spend about 99% of their lives below ground, and we have absolutely no idea what they do. And at a very, very brief period of the year, usually early January, they'll come up above ground and migrate down to depressions in the ground and lay their eggs and go back underground. And if the rains show up on time, those depressions will turn into temporary ponds. They'll pass through this very abbreviated larval stage that only lasts a few weeks, and then they'll go through metamorphosis, and just like the parents did, they'll go underground and we know nothing about them. So this is an example of a conservation partnership built to last. Example number 2, we're working with Japanese biologists way up in the mountains of Japan, working with the largest living amphibian on the planet, the Japanese giant salamander. There are old specimens, preserved specimens of this species that are 5 feet in total length. So we're not talking about a small amphibian. We're talking about the largest extant amphibian. This and the Chinese giant salamander are the 2 largest amphibians left on the planet, but they're in trouble. And a lot of the mountain waterways where they're found have been impounded, and there are small dams and large dams. But anytime you put a dam in, these salamanders can't get around the dam. And you can well imagine that if you take a waterway and you put 10 dams in, you've broken that population up into a whole bunch of little isolated populations. Any one of those isolated populations has reduced genetic diversity. Anytime you reduce the genetic diversity of a wild population, you are bringing into question the long-term survivability of that population. So working with our Japanese counterparts, one thing we knew we had to do was come up with a route wherein we could reconnect some of these currently isolated populations above and below impoundments. And so the Japanese biologists came up with this wonderful idea: why don't we put in salamander ladders that function just like salmon ladders do around dams? And in fact, we were able to help fund through San Antonio Zoo a study that the Japanese government required before any of these ladders could be put into place. Once that study was completed, the Japanese government gave the green light to putting in the very first salamander ladder. And that ladder was implemented about a year ago, and it is in place now. And in fact, salamanders are using it to get around a dam, and formerly isolated populations have been reconnected. So I use this as an example of a conservation partnership built to last, because there are entities in Japan that are involved, along with San Antonio Zoo's Center for Conservation and Research, along with the Japanese government, and along with a lot of these smaller Japanese cities and communities along these mountain waterways. Everybody's got to be working in unison to make these things last and work. And in fact, everybody's doing that. So the last example I'm going to show you of a conservation partnership built to last is the Chilean Amphibian Conservation Center. Now, before I get into the amphibian part of it, I want to talk about the habitat part of it. And if you look at this map, anywhere where you see green, those are the temperate rainforests of the world. And when I use the term temperate rainforests, what am I talking about? Temperate rainforests are just like tropical rainforests in that they receive 100 inches or more of rain a year. The difference is that temperate rainforests are in places where it's not tropical and it's not humid. In fact, they can get quite cold. It's one of the rarest habitat types on the planet, and the area of this map I want you to focus on is down on the southwestern coast of South America, along the coast of Chile. Unfortunately, that green area is what it used to be. Now there are only forest fragments left. What makes this habitat and this patch of forest so special is that it is entirely isolated from everything else in South America. If you go west, you're in the Pacific Ocean. If you try to go east, you hit the Andes Mountains, which are a high altitude barrier. If you go north, you hit the Atacama Desert, which is one of the driest places on the planet. So the vast majority of species, plants and animals, are only found in what now are forest fragments. So we got involved with the conservation of amphibians, primarily there because There are over 30 endemic amphibian species to South Chile in these forests. Only irregular conservation efforts were ever focused on 1 or 2 species. And emergent infectious disease was picking up because Chile is a developed economy, not a developing economy. And one thing that comes with developed economies is the pet trade. And so foreign amphibians had been imported that carried in disease with them, and that disease made it out into native populations in Chile. Unfortunately, deforestation and environmental contamination is on par with what exists in the United States. So when we got involved with this work, we had a lot of things in our favor. We have exceptional conservation partners in Chile, great biologists working with us. One of the things that I did to bias the success of this project early off, I grew up in a business where my dad imported aquarium fish from all around the world. Once he told his business partners that he had a kid that liked amphibians, little bags of gift frogs started showing up in the fish shipments. And oddly enough, as a 6 or 7-year-old kid, I was given a group of Darwin's frogs from Chile to work with through this system. This is in the 70s, before there were laws really regulating this kind of thing. And I taught myself how to breed Darwin's frogs in captivity, and actually had a colony that went for almost a decade. What I was able to do, because Darwin's frogs now are an endangered species, we started with Darwin's frogs down there because I knew I could breed them. In fact, once we got the frogs into the tanks, we had breeding activity within just a couple of months. And we had our first captive born baby Darwin's frogs produced in the following months. And that allowed us to get a lot of media coverage. And that media coverage changed our ability to fundraise for this conservation project. Going into it, we were only able to raise $25,000 or $30,000. After the media coverage, that pushed up to about $250,000. So the international interest in this project and the media coverage really made a difference. Any savvy conservation biologist simply has to pay attention to that kind of thing. We set up a massive exhibit at the National Zoo of Chile in Santiago, where we showed off some of these amphibians to the visiting public. We made one whole wall of that exhibit glass so that everybody could see it from outside of the lab. Now, Darwin's frogs are special for a variety of reasons. There are only 2 species of amphibians on the planet that enact their reproductive mode. Males will call on the forest floor to attract a female that's ready to mate. If a female is attracted to him, she will approach him, and all of a sudden she'll start jumping up and down on him, kicking him in the head, flipping him over, and beating him up. And if he's willing to put up with all of that, she'll go ahead and she'll lay a small clutch of eggs and take off. She is absolutely done with the process at that point. The male will fertilize the eggs and generally stay in the area until they hatch. As soon as those eggs hatch, the male will scoop the tadpoles with his hands into his mouth. He'll slide those tadpoles through an opening below his tongue called the vocal slit into his vocal sac. The vocal sac is the structure that frogs use to sing or make sound. And for the next 65 to 85 days, those tadpoles will develop within his vocal sac, within his body. It's a temperature dependent process. And then once he's done with that, once they're done developing, one at a time, he will cough those small frogs up into the environment. They hop off and they do their own thing. This is the very first captive bred baby Darwin's frog that was ever coughed up in one of our labs sitting at the end of my fingertip, to give you an idea of just how small they are. So one thing that's really interesting about Chile and all of these imperiled amphibians that I keep telling you about, and I told you that deforestation was on par with the United States. Well, here's what's really interesting about that. In the United States, deforestation had a lot of finance behind it, and even steep headwater valleys were deforested. In Chile, the finance was not there to cut out the forests and the steep headwater valleys. So if you're willing to ride horses and carry your gear and your supplies in on mules, and you're willing to ride for 1 or 2 or 3 days to get to these remote forest tracks, you can actually ride right into what Chile looked like 300 years ago, before any human devastation was unfolding with the environment. And these are very, very special places. These temperate rainforests are very old. The southern beech tree at the right-hand side of the image there was around at the time of the American Revolution. So these are very, very old, spectacular forests. And again, we're dealing with species found no place else on Earth. As you would expect with the rainforest, there are a lot of drainage systems. If you spend a lot of time in and around those drainage systems, you will find some incredibly rare amphibians. This species of amphibian is probably one of the rarest on the planet. It inhabits less than half a mile of one stream. That's all that's left of its habitat. So we will ride our horses out and carry our gear out with mules and spend time in these very remote forest tracks, taking inventory of the amphibians that are present. It's a great opportunity to get involved with Chilean graduate students and work with colleagues. Over the last 12 to 15 years, this has really been a spectacular project with which I've been lucky enough to be involved. One of the other regular activities we do, we monitor the amphibian populations in these remote forest tracts for emergent infectious amphibian disease. We're very, very careful moving between the tracks to make sure that we are not the mechanism that carries a disease between any two given forest tracks. And you can see I'm standing in a bucket of bleach there, cleaning off gear. So there are a lot of results to date from this project, and I don't want to bore you with all of them, but there's a key one at the very bottom of the slide where the Santiago Zoo and the Austral University have fully functioning captive breeding programs that are owned and operated and managed into the future by Chileans, not foreigners. And this is really an important point. If you want a conservation program to last, you cannot rely on foreigners to keep it going. There has to be local momentum. There has to be local biologists involved. There has to be local funding. Everything has to be local. If you cannot do that, these programs will not last. And that is the pathway and the template through which we build all of our conservation programs that don't take place on US soil. And again, the end game goal to all of this is to save some of these beautiful forest tracts that have species found no place else on Earth. This is a velvet worm or an onychophoran from Chilean temperate rainforest. This is an opilionid or a daddy longlegs, one of the species only found in these beautiful forests. And again, there are all kinds of incredible amphibians that really aren't found anyplace else on Earth. Just like the other projects, education and outreach, we've got a website just for this project, and it's savedarwinsfrogs.org for anybody that might be interested in a little bit more information. So I'm going to close up tonight talking about why you should care. And this is really an important point, and I know I'm preaching to the choir right now, but this argument is not being pitched to you. This argument is being pitched as such a thing that you can use later to convince folks that are not environmentally minded. And here's my two bits on this, because I've worked at institutions where I interface with the public for decades, and most of the folks come in and they're not really environmentally minded. I don't hold that against them. We don't teach environmental anything really well in the United States. It's a little bit depressing. So it's not surprising that folks come in completely unaware of what's going on in the environment and not necessarily even caring about it. It doesn't make them bad people. You just need to be able to sell them an argument that they can really connect with. So I start with the global amphibian crisis, and I talk about a mass extinction event. And we know that there is a long history of mass extinction events in the world, around the globe. We know that there were 5, and we are in the midst of the beginning of the 6th. The difference between 1 through 5 and 6 is that 1 through 5 unfolded by way of natural events. 6 is unfolding because of human activities. We are driving this extinction event as a species, and that makes it very, very different from the former 5. So why then can we, or how can we convince somebody who maybe isn't environmentally minded that they really should care anyway? And this is where I go with it. Humanity is losing its battle with antibiotic resistant bacteria. If you look at US congressional spending, we spend billions with a B every year on trying to develop new antibiotics for antibiotic resistant bacteria. When I use the term antibiotic resistant bacteria, I am referring to bacterial strains that no longer respond to standard antibiotics. So if you think about staph infections, right, caused by the bacterium Staphylococcus aureus. Most of them no longer respond to any of the first antibiotics that we used to use to effectively treat them. And the reasons for antibiotic resistance are really interesting. You can imagine generation time in bacteria is very fast relative to something like humans. So you, in just a few days, you can burn through many generations of bacteria. Well, if somebody is taking antibiotics and they don't finish the antibiotic regime, or they take it sporadically, what you end up with, if you end up with bacteria remaining, are bacteria that have whatever mutations that have allowed them to resist the antibiotics. And you can fast forward that through time and many hundreds of generations of bacteria. And it's not a stretch of the imagination to arrive to a place where you start dealing with bacterial strains that no longer respond to the antibiotics we used to be able to use to treat them. What does this mean in layman's terms and real world protocol? You can enter a hospital with a bacterial infection or get one in the hospital, which is a very common phenomenon, and there's nothing they can do to treat you. You will ultimately die from that bacterial infection. It happens every day in the United States, in hospitals and in convalescent homes. So these are rapidly emerging problems facing our society. We're all going to have to make a trip to the hospital at some point in our lives, or we didn't, so it doesn't matter to us anyway. But for those that did make it, and dealing with antibiotic resistant bacteria is a huge deal. And so now we're going to talk about the confluence of antibiotic resistant bacteria and biodiversity. And I want to go back to that group of animals that I've been harping on all night, the amphibians. And the amphibians evolutionarily played a huge role. That was the evolutionary lineage that made the transition from the water to the land. And that's a very, very special lineage because fish can't live out of water. They rely on gills. Gills don't function in air. So amphibians could make the transition from the water to the land, primarily because all living amphibians have a few characteristics in common. One of the characteristics that they have in common is a permeable skin. When I use that term, permeable skin, that means that they can, gases can freely pass through that barrier, as can water. So as long as amphibians keep their skin moist, they can breathe through their skin and they don't need gills. And in fact, a lot of extant or living amphibian groups don't even have lungs. There's an entire lineage of salamanders that don't even have lungs. They do all of their gas exchange directly across their skin. So back to this lineage that makes the transition from the water to the land, remembering that they have to keep their skin moist so that they can breathe through it. Let's think about this moist skin. What happens to moist skin in contemporary animals? Oh, that's right. That's where you get infections, terrible infections. That's where things like fungi and bacteria can really get going. It must have been carnage for the first waves of amphibians that came onto the land and died in mass because of these crazy bacterial and fungal infections that were developing on their permeable skin. Well, the evolutionary response in the amphibians, the mechanism through which they got around this problem, was the evolution of what's known as the granular gland. This is another characteristic that all living amphibians share. All living amphibians have these microscopic structures embedded in their skin that are called granular glands. You're looking at a cross section of amphibian skin on the right. Those circular structures that look like little embedded grapes are granular glands. What do? They fill with toxins and they contract, and those toxins are secreted out onto the skin of the amphibian, and microbes are kept at bay. Now, here's something really cool about amphibians. No two species have exactly the same chemical cocktail coming out of their granular glands. And again, to refresh from earlier in the talk, there are well north of 7,000 living species of amphibians on the planet. That's a lot of chemical diversity going on there. And that chemical diversity will come into play at this confluence of biodiversity and antibiotic resistance in bacteria. A lot of those brightly colored amphibians are brightly colored for a reason. They have very, very toxic skin. And those skin secretions, when we look at them biochemically, lo and behold, we can use them as biomedical advances in humans. So we look at pamiliotoxin, we're looking at that as a cardiac stimulant in humans. Epipetobetadine is being investigated as an analgesic. Maganin is being investigated and is already used as an organ glue and an antibiotic. Cerulean for intestinal ailments and pain relief, and adenoregulin for depression, treatment, and Alzheimer's disease. All of those things are in the works, and there are many hundreds of other studies looking at treating antibiotic resistance in bacterial strains with these very novel compounds that are produced by amphibians. Here's one other very, very special example. This particular frog is found in a little corner of Australia with very, very little of its habitat left. And it produces a chemical in its cocktail of skin secretions. And I say cocktail because they're not producing one or two compounds in their skin secretions. They're producing hundreds of compounds in their skin secretions. Only one of them is being discussed right here. But this compound X blocks mucosal transmission of the HIV virus with 100% effectiveness. So here's a critically endangered amphibian that's hanging on in a critically endangered tract of rainforest. And it is the only known production spot for this compound X that's being investigated that blocks mucosal transmission of the HIV virus. So quite literally, biodiversity is humanity's treasure chest for tomorrow's medicines. Don't take my word for it. Go ahead and Google and look up where ACE inhibitors came from. They came from the viper, from the venom of a viper in Brazil. Where did aspirin come from? The bark of a tree. Where did penicillin come from? A little green mold that grows on food that you leave too long in your refrigerator. The day before any of these biomedical advancements were discovered, those species had absolutely no value to most humans on the face of the planet. Think about the number of people you know that are on ACE inhibitors. They owe their lives to a little viper that lives in South America. So this is where humanity's next medicines will come from. I want to make a point here, and it's a really important one. My personal belief system does not require boiling biodiversity down to what humanity can get out of it. I believe that every species has just as much right to be here as we do. But if we're dealing with pitching an argument to someone who is not environmentally minded, where their next medicines are going to come from, where their kids' medicines are going to come from, and their grandchildren's medicines are going to come from, that might catch their ear. So now what I want you to do is insert any group, biological group, and chances are the story is exactly the same. If we're talking about tropical birds, if we're talking about earthworms around the world, if we're talking about corals living out on a coral reef, if we're talking about exotic plants from some distant corner of the planet, these groups more than likely are suffering from the same environmental issues that amphibians are suffering from. Most groups of animals face the exact same challenges, but all is not lost. And this is a really, really important message that I want all of you to leave here with tonight. There are still so many places worth fighting for. This is a photograph I took out of a small bush plain in the Amazon basin. I have camped at this bend of the river, and it is a spectacular rainforest. I sleep in a hammock and I listen to the jaguars roaring at one another from across the river all night long. This place has most or all of its biodiversity still intact. They are so special, and so many species are still with us. I know a lot of times people take these discussions into really negative places and, oh, we've lost too much and too much is gone. That is absolutely not the case. Now is the time to step up and fight for these things, not back down. The forests still have their wildlife, and the wildlife can still be experienced in person. We can take people there. I take groups of folks to the Amazon to see those places and experience the biodiversity firsthand. You can still do that. These things still exist, and there's so much wildlife we can still learn from. So many venomous species are producing medicines that can be used to fight human diseases. In fact, one of the leading treatments of breast cancer in humans comes from the southern copperhead, and that is a species that's found right here in South Texas. And the one component of its very complicated cocktail of compounds in its venom is being investigated as a potentially a very, very effective treatment for breast cancer in humans. Again, another species that the day before this research started, nobody would have cared about. And now a lot of folks care a lot about it because I think we probably all know somebody who's been impacted by breast cancer. So here's the thing. We don't get to pick and choose which species might be of benefit. And I want to talk about something called technological constraint. I talked about this in my last book, Life in the Dark. If you think about contemporary technologies and you think about even 10 years ago, how simple cell phones were versus what we have today, technology is advancing at unprecedented rates. Tomorrow's technology will undoubtedly allow us to look at wildlife in ways that we couldn't possibly even fathom right now. So you can't ever look at a species and say it is without value, because tomorrow's technologies might turn that on its ear. And there are plenty of examples of that. When technology got just sophisticated enough to look, lo and behold, there were things there that we didn't see from our very simplified eye. So technological constraint guarantees that we can't say any species is without value. Therefore, our default has to be the preservation of all biodiversity. We don't get to pick and choose where the next biological development might come from. So that's my pitch, and I hope it'll help you sometime sitting down with friends that are maybe not environmentally minded or family members. But biodiversity quite literally is attached at the hip with humanity. If biodiversity fades, so will we. There is no question about that. So I hope in the talk tonight, I've delivered a few key take-home messages, particularly with amphibians. So time is short, and we need to really investigate how we can protect amphibian biodiversity because it's disappearing. 35 to 50% of all of the species are in some form of trouble. No one is arguing that the methods we investigate today will solve amphibian decline. But if we don't wade in and look at each one of these pathways, we're not going to know where to start. Developing capacity to accommodate assurance colonies of the most endangered of these things is definitely a stepping off point. I hope that I've convinced you that securing a future for wildlife at San Antonio Zoo's Center for Conservation and Research, that we have boots on the ground. Most of our field work is grant foundation and donation funded, and we have clear program direction in everything that we do. I just want to emphasize the group of people watching this tonight. We have to fight for the preservation of biodiversity before the sun sets on it, or what we're going to do is one by one watch these species vanish. And that's something that we can't ever fix. Once it's gone, it's gone. If you want to know how you can help, go to SAZCCR.org, SAZOO.org. Click on the conservation button in the upper left-hand corner. You can read more about our conservation projects, and there are donate buttons all over the place. Thank you so much for your time tonight.
This is just fantastic. This, I don't know what to say. And I don't know what to say about our technology. Very embarrassing.
No, we're
But-
fine. We're fine.
Okay,
Yeah.
but do we have any questions? Oh, yeah, we got to end the recording.
There are a lot of questions on the chat.
Okay, well, we can.
Well, do you have any voice left to do
Oh, yeah,
that?
I'm fine. I'm
Okay.
fine. Yeah, this is kind
Okay.
of typical.
I don't know how to do it. Participants chat.
Can we end the recording?
Oh, my pleasure.
Amazing.
No problem. Thank you.
Wow.
Thank you. Yes, ma'am.
I'm sorry, I didn't